Do you want to do both of them on the same one?
I don't know if we can.
We can just post the same YouTube link.
Right? Okay, sure.
Yeah. So what we're doing today is we're going to do both lesson 10 and 11 applying group problems. We've both worked ahead a week since we're both are getting out of town, so we don't have time to work anything next week. So we'll post both this video for both weeks, lesson 10 and 11. So we'll just do it all in one video. Okay. Let's go ahead. So lesson 10. Do you want to start or do you want me to?
Let's see. I don't really quite have the problems opened up yet. So why don't you go ahead and start while I go look for those problems, okay?
Okay. So for question 1, translation in e-coli is subdivided into 3 stages: initiation, elongation, and termination. So what mRNA sequences, rRNA molecules, and tRNA molecules are involved in translation, initiation, and E. coli? So I said it's the Shine-de-la-Garneau sequence, the start codon, 16S rRNA, and initiator tRNA. For that one. So for B,
huh? Could you repeat it one more time? Sorry, Joachim.
The answer?
The answers, yeah.
Shine-de-la-Garneau sequence, start codon, 16S RNA, and initiator tRNA.
Right. Or, I wrote the specific tRNA, like I wrote the tRNA F methionine.
Oh, okay.
It's the same thing, though, basically, right?
Yeah. Yeah.
Okay.
So what proteins are involved in translation initiation in E. coli? And that's the IF3 and IF2.
Yep.
And what mRNA sequences?
I had a question. I had that question about Part B. Would the ribosome subunits be involved in that answer? Because technically they are proteins.
Let's see.
The 30S subunit and the 50S subunit.
Yeah, I mean, that's a good question.
So, I mean, it wasn't in the key. I know that.
You're right. You're right. That's true.
But they are technically proteins. Like, they're made up of rRNA and proteins. So.
Yeah. You're right. I don't know.
Yeah, I don't know either. So, I mean, I guess the important thing she wants us to gather from it is what proteins exactly are involved in helping combine the ribosomes together over the RNA, kind of sandwich over it, and that would be the initiation factors 2 and 3.
Okay.
So, I mean, in all actuality, yeah, technically, like it would be the ribosomes, but I think the real important thing is that we get the IF2 and IF3. So
yeah.
Okay.
So what mRNA sequences and the rRNA are involved in translation, elongation, and E. coli? So let's see. See, that's the codons on the mRNA. They are there, and the 16S rRNA and the 23S rRNA.
The 23S rRNA, that's the ribozyme that combines each amino acid together in a peptide bond, correct?
Let's see. Let me look real quick. Yep. Woohoo! That is correct.
Whew. Okay.
Okay. Let's see. Where are we? D.
Uh-huh.
So, what proteins are involved in translation elongation in E. coli? That's F2 and FG. EF. EF2 and FG.
EFG. Okay.
So, then what mRNA sequences and proteins are involved in translation termination in E. coli? A stop codon, RF1, and RF2. So, no questions there, but don't you think we should just like maybe focus on the things there where we maybe were like, or do you want to go through the whole thing?
No, it's up to you, man. Like,
Or
uh,
I mean, we could just focus on like the things where we had questions, or
yeah, I
were
think,
like unsure why.
yeah, the thing is though, I didn't have too many questions on this. Like, all of it was
Mm,
pretty straightforward to me. Yeah.
yeah. Um. Let's see.
Oh,
Three.
excuse me.
Three was just translating amino acids. Two, that was just making. Yeah, that was also like the wobble rules. It wasn't too hard.
Right.
Um.
These are pretty straightforward stuff, if you ask me.
So the energy. Let's see. Um. So how many ATP? One ATP to charge a molecule. Initiation step does 2 GTP and elongation does 2 and 1 for termination, right? So through
Yeah.
a whole translation, it's 5 GTP and 1 ATP then, or what?
Well, you got to remember though that like when it's doing elongation, it's
Oh, overdoing it twice, like
yeah, two GTP molecules every
right,
cycle, and
right, and
then
then
every single time a tRNA is charged, that costs one ATP.
okay,
So it uses a ton actually.
right. So then for 5, uh, what you had to look at was to like write the sequences as the wild tab one. I mean, it could be different ones, but yeah, like
Yeah, that was the thing on 5 was that um, the answers could vary, right? Because it says one of the possible mRNA sequences.
mhm.
But doesn't that kind of like change the rest of your answers, like B through D or B through C? I mean,
I suppose it could.
or rather, or sorry, rather B. So
Yeah,
I don't
but
know.
they wanted it to like notice what happened since like that deletion occurred in like D, I think. Sorry,
Hmm.
but I mean,
Yeah, I think that, like mine worked, I think, but
yeah.
like I had to like do a couple of additional like. Did you only have to do one change on each mutant?
Um. Well, yeah. I So B, that was like a for a G or
Yeah,
G
it
for
just
an A. Like it's made a stop codon or something like that.
it code, yeah.
And the C, uh, it
That's just
actual what the last one was, and that's a stop codon. And then in the D, uh, oh, what sequence is contained? Oh, so that was like the what do you call it? Shandagado sequence.
Yeah, Shine Together sequence.
Okay. I feel pretty done on that one.
Yeah, it sounds good to me.
Let's go to lesson 11, teach one another.
Yay. Okay. Well, I will do this one. Let's see. Okay, so it asks us what are the two regulatory proteins that are involved in the lac repressor, or lac operon? Sorry, lac repressor is one of the answers, and then the catabolic activator protein, or CAP, in other words. Those are the
Yep.
two regulatory proteins.
Yep.
And then what are the two effector molecules of the in-regulated lac operon? That'd be cyclic AMP or CAMP, and allolactose.
Yep.
Then in C, it says what 4 DNA sequences are involved in regulatory transcription of the lac operon. That would be, I actually wrote a number of answers. Because, you had the operator, the promoter, so lac O, lac P, and then the cap site, where the cap would bind. And
It
then
says
the lac.
regulating, though.
Yeah, does the cap site not count?
But the cap site is there because that's a regulating one. The lac promoter is in the operator site and the lac terminator. So like the genes are taken out.
Yeah, because they don't regulate it. They're not
No.
involved in regulating it.
Nope.
They're the things that are being regulated, you know.
Yep.
But I was wondering why lac I wasn't involved in that.
Lac I.
Yeah, lac I is
it's not in
before.
lac I is not part of the operon.
It's not. It's not considered part of the operon.
Nope.
Oh, well, that stinks for lac I. It's like that lonely kid who eats alone at lunch. Okay. What 3 structural genes are found within the lac operon? That'd be lac A, lac Y, and lac Z.
Yep.
Drop the mic. Okay. In 2, the first part asks, what happens to the expression? Suppose we isolate an E. coli strain that produces an inactive lac repressor protein that cannot bind to the operator sequence of the lac operon. What happens to the expression of the lac operon genes? So if it has an inactive lac repressor protein, then that means this protein is pretty much always going to be on, right?
Yep.
Like it's just going to be like on forever because there's nothing that's stopping it. Then it asks if it's advantageous or disadvantageous. So I wrote disadvantageous because if they're spending the energy to make these enzymes and there's no lactose to like break down or anything like that, then there's no point in it and you just wasted energy. So it's an energy efficiency thing.
Yep, that's true.
Okay, 3. Okay, 3 was a little bit more confusing for me, particularly what the difference between inducible, non-inducible, and constitutive are. Okay, so it says in the lac operon, how would gene expression be affected if one of the following DNA sequences were defective? So for the lac promoter, if that was defective, then that means the RNA polymerase. Say again?
Yeah, you're right.
Yeah, the RNA polymerase wouldn't even be able to bind to it and do transcription.
In all cases, it won't ever work.
Yeah, pretty much.
Inducible.
So yeah, non-inducible. Just does non-inducible mean that like it can't be induced to start transcription?
Yep.
Okay, well, all right. Then in B, it says the operator site. You know, if the operator site was defective, then that means
it can't bind. Yeah, what? It will never be inhibited.
Yeah, it will never be inhibited. Exactly. So it's constitutive because that means it's on always, 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 right?
Mm-hmm.
Now the lac Y gene. Now I didn't get this at first, but like after reading the key, it made more sense. But I didn't realize that I kind of thought that since the LAC-Y gene was being transcribed, it didn't matter. But it turns out it does, because if the LAC-Y gene
It's the lactose
permase.
permase that brings
Yeah,
the lactose there. So right.
exactly. Yeah. So it's the thing that brings from the outside of the cell into inside
So
the cell.
lac repressor would just stay on the oppressor.
Pretty much, yeah. It would be non-inducible again. And, like, there would be no lactose coming inside. And in LAC-Z, I realized LAC-Z makes beta-galactobase, which turns lactose into allolactose.
Mm-hmm.
So it would be non-inducible on part D, because like you wouldn't make any beta-lactamase, so you would have lactose, but like it couldn't bind to the repressor because it's not allolactose.
So this lesson, I was like sitting today and I actually did it all today.
Oh,
And I
okay.
was sitting there like when I first started reading, and I was like, this does not make sense at all.
Yeah.
And then I read it, and then
Have you taken microbiology yet?
No. Well, then
Yeah.
I went and watched the video, did the reading a lot, like more in depth, and I get it pretty good now.
Sweet. That's what I'm talking about.
And then I've done both of the applying and teaching one another, and the quiz.
Yeah. And the quiz and everything. So, yeah, like the idea, like they always use the LAC operon as an example of like regulating sequences. Like you learn about the LAC operon like so often that you pretty much like, I don't know.
I actually figured that. It's like those videos, everyone talked about the LAC operon and the TRF or some Trifone or something like that.
Tryptophan, you mean?
Yeah, that was the other one.
Yeah, tryptophan is the other one, which is basically like the opposite. Like the LAC operon uses a repressor, the tryptophan an activator.
Yeah, that one comes in and binds when there's a lot, right? And then where there's
that's right. Less. Yeah, it comes in binds because like it's its way to tell if it's like it binds if there's a lot, then it binds the repressor to it. If there's not very much, then the repressor is off. So it's kind of the opposite, sort of. Okay. So the part of this lesson that kind of threw me for a loop was this Jacob Monday Pardee experiment.
Oh, yeah.
I think I get it. So, for example, it says why was beta ONPG used in this experiment? Basically, that's kind of like the substrate for the beta-galactamase. That if there was beta-galactamase on there, it would react with this and it would turn the solution yellow.
Right.
So
it's to see if it got transcribed.
Pretty much, yeah. It's basically to see if it got transcribed under varying conditions. Part B asks, why was there no yellow color observed in one of the 4 tubes? Well, one of the 4 tubes lacked any lactose, I think, right?
I just said it was turned off.
Yeah, it said the tube containing no lactose and the merozygote strain of E. coli had the genes always repressed. So beta-galactamase was not produced and beta-ONPG was never cleaved. So no color change, basically. Like, it's basically what you said. Like it's always turned off because it's always being repressed. So, what is another method that we could use to measure the level of expression of the LAC operon? I said try cyclic AMP instead, but I think the key suggested using one of the other.
I mean, I'm sure there's a lot of answers you could use there. That was a hard question, honestly. Like,
yeah, that was a hard question, and I was like, oh, okay.
And I'm also like, analyze everything you've ever learned in anything.
Exactly. Use different cells. Use a different substrate. Like, there's a lot.
I like the idea of what they said. Like to use, what was it? Like isotope of lactose or something like that to see what will happen to it.
Yeah. They said something else, but I can't remember. Anyway, that's not important. Yeah. So in other words, C has a
So,
lot of.
like,
Yeah. Use your scientific, intellectual part of yourself to come up with a good answer and impress the teacher.
pretty much.
Okay, part D says, why was the optical density twice as high in tube 4 as in tube 1 and 2? Well, the reason why was because tube 4 had a merozygote, which basically has double the genes, and
Mhm.
both were, let's see, one was always turned on, and the other one was like a normal one. So, so the one that was always turned on, of course, would produce beta galactamase, which therefore would react with beta ONPG, make its normal yellow color. But the second one also was turned on, but that's because the repressor was released from it because it was in the presence of lactose. So you had twice as much. It's pretty much just you had double the genes, double the power.
Right. Yeah.
Yay. 5 is a little difficult. Once you go
I thought that one was easy.
Oh, did you? Okay.
Yeah. I thought this one was super easy.
Oh, all right.
It's basically, it says super simple idea. Like glucose, if that's present, it inhibits adenylyl, what's it called? Hold on.
Lactose or allolactose?
No, adenyl cyclase.
Oh, I mean cyclic AMP.
So it doesn't cleave ATP to cyclic AMP, right?
Mm-hmm. That's right.
So if that occurs, so if glucose is there, there's going to be low levels of CAMP, whereas if there are low levels of glucose, there's high levels of cyclic AMP. Cyclic AMP is sort of like an activator, so if that's there, it's going to initiate transcription, and then you just have the lactose. If lactose is present, it's going to take off the repressor. If it's not present, it's not going to take off the repressor. So for it to occur, lactose has to be present and glucose cannot be present. That's
exactly.
Huh?
Sorry, I was going to say, so pretty much the only one that really works is C. C is the only case where it's actually transcribed. All the other ones are not transcribed.
Yeah. So we pretty much went through that now. The whole thing.
Yeah, pretty much. Pretty much.
Cool. Cool.
Yeah.
So let's meet again in what, like 2 weeks? So I think in 2 weeks, that's like the last lesson. Then it's just like a final.
I think so, or something like that. Do we finish like a week earlier than the actual semester?
I don't know, but that would be great. My math class has like 3 assignments, 3 days in that week. But I mean, it's because we have the final. Usually like the school classes ends on like Tuesday or Wednesday or something like that.
Yeah, it's like Wednesday or something like that. And the last 2 days are like
it just seems like everyone's gone the last week. I don't know why. I still have classes.
Yeah, it's because everyone has easy majors, dude. Like everybody is always finished by like the second to last week, and they're out of there.
I know. I'm like, I get done on every time, Thursday or Friday. Like,
Me too. I'm like, I can't like leave any earlier than that, man. Like that's when I can actually get, because I usually have like 5 or 6 tests the finals week.
Yeah,